We are going to start the session of today on, this is technical session 7 on One Health and Aquatic Animal Biosecurity. We have for today the chairpersons, Dr. George Ninan and Dr. Andrew Shin. I welcome them and I invite them, the chairpersons, Dr. Andrew Shin and Dr. George Ninan will be joining very shortly to kindly come over the stage. Dr. Shin. And for today's presentation, we have keynote presentation by Dr. Idya Karnasagar. Dr. Idya Karnasagar, kindly come over the stage. And we have second keynote presentation by Dr. Melba. Dr. Melba, you are kindly invited over the stage. And then third presentation is the lead presentation by Dr. Victoria Alde Sanz. Dr. Victoria, please. For this session, we have coordinators, Dr. S.K. Ota from ICR SIBA and Dr. Samantha from ICR SIFA. I welcome them also. And I request the session coordinators, Dr. Ota and Dr. Samantha, to kindly coordinate the session, please.
Thank you, Dr. Sekhar. Good morning. And a warm welcome again on the last day, the 26th of September, the first session, the technical session 7, that One Health and Aquatic Animal Biosecurity. So in this session, as Dr. Sekhar mentioned, there are two chairpersons, Professor Andrew Shin and Dr. George Ninan. So these two distinguished chairpersons, let me introduce our Professor Andrew Shin. Professor Andrew Shin, a global technical expert at Benchmark's Advanced Nutrition, specializing in disease management, with over 35 years of experience spanning diagnostics, biosecurity, and welfare. Professor Shin has made transformative contributions to aquaculture across Asia and beyond. He is the co-founder and overseas director of the Fish Museum and Biodiversity Center, Mymensingh, Bangladesh, and is an invited member of the Network of Aquaculture Center in Asia Pacific, NACA, Asia Regional Advisory Group on Aquatic Animal Health. He has a PhD in veterinary aquaculture from the Institute of Aquaculture, University of Stirling, Scotland, and the Natural History Museum, London. A prolific researcher, Professor Andrew Shin, with more than 190 peer-reviewed publications, he also holds honorary academic positions in the UK and Australia, and actively advises international bodies, including FAO and OIE. So, Professor Andrew Shin, we welcome you to these sessions and request you to chair this session. So, Professor.
Good morning. So, the organizer is also pleased to introduce our next chair, Dr. George Ninan. So, Dr. George Ninan is the director of the ICR Central Institute of Fisheries Technology, Kochi, and a nationally recognized expert in fish processing technology. With a distinguished academic background, including an MSc in Industrial Fisheries and a PhD in Marine Science from the Cochin University of Science and Technology, Dr. Ninan has been at the forefront of innovation in aquatic food processing and sustainable marine resource utilization. He has led several significant projects, including ICR-funded programs on standardization of novel aquatic products, shelf-life modeling, energy assessment, and value-added byproducts. He was also the principal investigator of the Zonal Technology Management and Agribusiness Incubation Unit of ICR-CIFT under NAIF and contributed to a CMLRE-funded project on microfeed resource assessment and development of post-harvest technology. Dr. Ninan's contributions have earned him several accolades, including the Jawaharlal Nehru Award for Outstanding Doctoral Research and the A.V. Jones Young Scientist Award. He has authored several impactful scientific publications, especially in the area of fish minced-based products and fish gelatin. So with this, I request the chairs to continue the session.
A very warm and good morning to everybody. It's lovely to see so many fresh faces in the audience. This morning, I think we've raised the game. We're not having just 2 key presentations, we actually have 3. So it's absolutely my pleasure and privilege to be here to introduce our first speaker, who almost needs no introduction because I think he's a friend and known to us all, but I would like to give him an introduction. So our first keynote speaker this morning is Dr. Idya Karunasagar, who obtained his master's and PhD degrees in microbiology from Mysore University in India. And he carried out postdoctoral research at the University of Maryland in the States, and also at the University of Sendai, Japan, and Natural Resources Institute in the UK, and also the University of Würzburg in Germany, where he was a Humboldt scholar, and also a visiting professor. He joined the University of Agricultural Sciences, College of Fisheries, Mangalore, as an assistant professor, and rose to become the professor, and then the head of division of fishery science and the director of research at Karnataka Veterinary Animal and Fisheries Science University, Indian Council of Agricultural Research, who awarded him a national professorship. He then moved to the Food and Agricultural Organization, FAO, of United Nations, and was based in Rome as a senior fisheries officer, where he led a team on fish safety and quality, and was based at FAO headquarters in Rome throughout that period. He then played a key role in several FAO and World Health Organization food safety risk assessments, which led him to the development of international food standards at the Codex Alimentaris Commission. He represented FAO in meetings of Codex Committee on Food Hygiene and Codex Committee on Fish and Fishery Products. He was the lead technical officer for several food safety projects in Asia, in Europe, Africa, South Pacific, Caribbean islands, and Latin America, so very, very extensive. He played a pivotal role in the development of the World Health Organization 5 Keys for Safer Aquaculture Products. And then in 2021, he was appointed as a member of the World Health Organization Advisory Group on critically important antimicrobials for human use, and he is a member of the World Health Organization International Team for Source Attribution to Food Diseases. He is presently working as a research advisor at NITI University, Mangalore, India. And with that, it gives me the greatest of pleasure to invite you to the podium and to deliver your keynote speech, sir. A warm welcome, please.
Good morning to all of you. Thank you very much, Andy, for this very kind introduction, and thanks to Dr. Kuldeep for the invitation and also for the excellent organization of this conference. So this talk is a joint presentation from me and Melba. So Melba is going to talk a little bit more on One Health. I will talk on the AMR part in the first part. Now, as you all know, AMR, so AMR is an important aspect now. Everybody is talking about AMR in all fields, in the medical field, in the veterinary field, also in aquaculture, and it's getting a lot of attention. And this is one area which fits very well into this concept of One Health, where we say the human health, animal health, and environment are closely interrelated, and you cannot address any problem in any of the sectors individually. You will also always have to look at other sectors and see how we can address them together. And Melba is going to talk more about One Health later. So this figure came from one of the articles which I published in the FAO Fisheries and Aquaculture technical paper, where I tried to highlight, because people talk about AMR in aquaculture, and you say, if you look at the water body, any water body, you will find resistant microorganisms. And always the thinking is, if you find resistance in a particular environment, that means there has been use of antibiotic there. But this is not true, because particularly aquatic environment receives effluent from all fields, from the hospitals, from animal farms, from agriculture. So whenever antibiotics are used in any sector, ultimately the drug residues end up in the aquatic environment, and also resistant microorganisms that have arisen either in the hospitals or in animal farms, they finally end up in the aquatic environment and will reach aquaculture. So what we find in the aquatic environment, resistant organism might have come from any sector. So it's, researchers have to be careful while making conclusions. There was way back one paper from Australia where they looked at some water body and they found some resistant organisms, and they said, though it is not a permitted antibiotic, since the resistance is there, there must have been some unlabeled use. So, but this is not the case, because you will find resistance in almost all part of aquatic environment. So we need to be careful while concluding about the source of the resistance. Now, when it comes to food safety, now does this resistance associated with the antimicrobial, the aquatic environment, does it affect the humans when you consume fish? So does it affect humans? So we need to look at what are the pathogens that can be transmitted to humans through food. Now, if you look at the records of foodborne infections associated with seafoods, it's much less compared to many of the other products. It's generally a safe food, but if you look at the statistics, see, many of the outbreaks have been related to some toxins which have been associated with fish, or due to bacteria like salmonella, which come from other sectors. So there are very few organisms that are actually innate or native to the aquatic environment. So here you have a list of organisms, some of which are native to the aquatic environment, like Vibrio parahemolyticus, but not all Vibrio parahemolyticus. There are certain strains which are positive for TDH and TRH gene. Then Streptococcus agalactiae now is getting more importance. Vibrio vulnificus, which causes infection, particularly in bivalve consumers. Clostridium botulinum in canned products. Then there are organisms derived through contamination, like Salmonella, norovirus, hepatitis. Listeria, also we include under this category because it comes from environmental contamination. It is there in the environment. So these are the main organisms, but I will try to focus on two organisms, and we'll look at how resistance is coming up in this and how pathogens are emerging in this sector. So first, talking about Vibrio parahemolyticus. Now, Vibrio parahemolyticus has been known for a long period of time. So in fact, my career is very much associated with this Vibrio parahemolyticus, because when I joined the College of Fisheries in 1978, my first project was on Vibrio parahemolyticus. That time it was known only as a foodborne pathogen, and countries were looking at this organism to see whether it can be a fish-borne hazard. And even European Union was thinking of having some limits for the levels of Vibrio parahemolyticus, because particularly in countries where they consume bivalves raw, then there are cases of Vibrio parahemolyticus. So Codex was thinking of developing standard for Vibrio parahemolyticus, then FAO and WHO through the joint FAO and WHO committee for risk assessment came up with these two risk assessments. And that indicated that the levels of Vibrio parahemolyticus in different geographical regions are different. So you cannot come up with any level that can be prescribed in terms of standards. Then Codex came up with a guideline on how to control this Vibrio parahemolyticus in bivalve mollusks and other seafoods that are consumed raw. But now in 2011 or 12, we had this problem of early mortality syndrome, and then it came out that Vibrio parahemolyticus can also be a pathogen of aquatic animals. So on one hand, we have these human pathogens, and they are categorized by the presence of certain genes. So even though this organism is present in the aquatic environment globally, wherever temperatures are more than 15 degrees, but you will find that most of the environmental strains are not pathogenic to humans. there are only a very small percentage, maybe about 3 to 4%, which are actually pathogenic to humans. And they carry some specific genes coding for hemolysins, thermostable direct hemolysin, TDH, and TDH-related hemolysin and type 3 secretion system. And then environmental strains which are negative for these. And then we came to know that there are some shrimp pathogenic strains which emerged in around 2009, and then of course I think it was identified only as in 2011 or 12 as the causative agent of the early mortality syndrome, or AHPND. But you can see here the emergence of the pathogen, because this AHPND organisms have a plasmid. So the virulence gene is carried in a plasmid which contains PIR-A and PIR-B genes. So they are negative for TDH and TRH, which are present in human pathogens. So because that time there was a lot of concern when there was so much of outbreak of AHPND, can we consume shrimp? Is it harmful to consume shrimp? Then of course the data showed that these shrimp pathogenic ones, they lack this TDH and TRH genes which are required for human pathogenicity. And then of course now we have one more pathogen. Here also we had oral presentation and also poster on this translucent post-larval disease, which is also caused by Vibrio parahemolyticus. But this possesses another plasmid, a mobile genetic element, which contains genes coding for this hypervirulent protein, VHPVP1, 2, and 3. So this is carried in a large plasmid. So now we have three categories of this Vibrio parahemolyticus in the environment. Because if you look in the aquatic environment, shrimp ponds, you will always find them. But are they causing AHPND, or are they responsible for TPD, or are they environmental strains we need to look at? So this shows the evolution of this Vibrio parahemolyticus, where you can see they acquired different types of genes before they became human pathogenic. So they acquired the type 3 secretion system and the thermostable direct hemolysin, and then from the environmental strains which were negative for this TDH, TRH, and also for the type 3 secretion system, we have the emergence of the HPND strain, which carried the plasmid for PIR-A, PIR-B, and then the translucent larval disease strain, which is carrying another plasmid. So here you can see, just to illustrate, that this is a mobile genetic element. So the genes are acquired by horizontal transmission. So you can see here PIR-A-B genes in the plasmid flanked by transposase. So transposons are involved in mobility of the genetic element. So these are mobile genetic elements, so they can be transmitted. That's why AHPND can be caused not only by Vibrio parahemolyticus, but other Vibrios which acquire this plasmid. And then the causative agent of translucent larval disease. So this has the large plasmid of 187 kb, plus it has a type 4 secretion system which allows the transfer of the pathogen into the another organism by conjugal means. So again shows how the mobility can happen and how the organism can acquire genes and become pathogenic. And if you look at the AMR associated with this, it varies regionally. So in the case of China, they have found large number of strains which are not so resistant. And then if Vietnam, there are some papers related to resistance which shows that it varies geographically. So it may vary in different environments depending on the exposure. And then of course there is a lot of genome resource which one can use for studying antimicrobial resistance. So another organism I just want to highlight is this one now which has emerged recently, the Streptococcus B, Streptococcus agalactiae, particularly the sequence type 283. And this was known as a fish pathogen long back, but only in 2015 it was identified as a human pathogen causing foodborne infection. So here the sequence type 283 is the one which is causing human infections. And of course I don't want to, and a lot of genomic resources are available to study this. And resistance is still very small in this Streptococcus agalactiae, particularly 283. So it's unlikely to cause infections due to resistant organisms. So a lot of resources are available. And will these resistant organisms cause human infection? Now, they have been causing infection. So what is the characteristic of a AMR infection? So it should be either a treatment failure or prolonged hospitality. So how to understand whether these are coming out of these pathogens? So Codex has this guideline and how to perform this risk assessment. So we need to follow this methodology to understand whether these resistant organisms are causing human infections. Thank you.
Thank you very much. We do have time for a quick question, if anyone would like to, if we have any questions from the floor. Yes, we have a question at the back.
Thank you, sir, for the wonderful insight. I'm from a quarantine facility of our country. Would like to know about the transmission mode of TPD. So whether it is a vertical or a horizontal transmission. Could you please throw some light on that?
Yeah, of course, I think there is no evidence that it can be transmitted vertically because it's a bacterium. So it's a Vibrio parahemolyticus. And of course, yesterday there was presentation about EHP. Even EHP is not transmitted vertically. It's all horizontal transmission. So this bacterial transmission is horizontal, not vertical. So it's unlikely to be transmitted to the next generation.
Okay, thank you, sir.
Thank you, sir, for your wonderful presentation. And actually, it is I wish to ask one question that is just for my learning and understanding sake, that Parahemolyticus, actually it is close to many other species. But what I find that compared to all its related species, it is evolving pathogenically much faster than others. What could be the reason which you feel that why it is evolving so fast compared to that other related organisms? They all also have all the transposons and mobilome, but Parahemolyticus is evolving much faster.
Yeah, of course, we know more about Vibrio parahemolyticus because we studied it more. So we have not studied the other organisms as closely as Parahemolyticus, maybe because initially it was understood as a foodborne pathogen and now as a shrimp pathogen. But where do these genes come from? So the genes, now for example, AHPND plasmid came from some other organism, maybe from Photorhabdus. So it is acquiring genes. So this type of process has been happening in nature. Only thing is we have not looked into them. We are not studying organisms which are not pathogenic. So that's why we don't know much about other organisms. So it is happening in nature. Other organisms are also acquiring plasmids. And of course, Parahemolyticus belongs to the group of organisms, Vibrio, Harvey, clade. So there a lot of genetic exchange has been taking place. So it is happening in other organisms also, but we know more about Parahemolyticus because of its importance for commercial aquaculture and also from human health.
Okay, thank you very much. Again, if we could just give a warm thanks to Professor. Thank you.
A very good morning to all of you. Friends, I have the pleasure to introduce the next speaker, Dr. Melba G. Bodant Reintaso. Dr. Melba is a globally recognized expert in aquatic animal health and biosecurity with nearly 30 years of experience spanning research, diagnostics, extension, and international development. She currently leads the food safety, nutrition, and health team at FAO Fisheries and Aquaculture Division, where she spearheads global initiatives on disease surveillance, AMR, and biosecurity, including progressive management pathway for aquaculture biosecurity. She spearheaded the development of a new initiative called Progressive Management Pathway for Improving Aquaculture Biosecurity, a paradigm shift in managing health of aquatic species through a risk-based, collaborative, and progressive approach. A former senior aquaculturist in the Philippines and a regional specialist at NACA, she has led investigations across 70+ countries and contributed to over 50 FAO projects. She holds advanced degrees and postdoctoral training in aquatic animal diseases from institutions in Japan, Canada, and the Philippines. Ma'am, the floor is yours.
Good morning, everyone. So I am going to present the second part of this keynote presentation, focusing a little bit on One Health and application of One Health AMR in the aquatic sector. So how do I do it? Okay, so just to give you a little bit of the history of One Health. So in the 19th century, the One Medicine thinking came about to jointly address animal and human health issues. And actually, this came from veterinary doctors. And fast forward, in 2004, the One World, One Health thinking recognized the link between human, animals, and the environment, and emphasized the need for interdisciplinary approaches to prevention, investment, policy development, and education. In 2006, amidst the avian influenza scare, the One Health Initiative Task Force was formed. And in 2017, the tripartite organization collaboration between FAO, WOA, and WHO was established. The COVID-19 pandemic re-emphasized the need to work closely together using an interdisciplinary approach to prevent, invest, develop policy, and education. So in 2029, we now have the One Health definition. From the tripartite collaboration that was established in 2017, it has now developed to a quadripartite collaboration, including UNEP. Okay, so this is now the definition of One Health. It is an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. It recognizes the health of humans, domestic and wild animals, including aquatic, plants, and the wider environment, including ecosystems that are closely linked and interdependent. So you will see here that, can you see the little fish here? Maybe I'll use another. Can you see the little fish here? So we are important. We are part of the sectors and disciplines, and we are contributing to the creation of healthy ecosystem, healthy animals, and healthy humans. The four pillars that supports the One Health is 4 C's: communication, collaboration, coordination, and capacity building. Okay, so I got this from the WHO website. These are the One Health application. I will not go into much detail because in the interest of time, but it's AMR, zoonotic disease, that we will deal with later on in my presentation. Dr. Karun already covered AMR, vector-borne diseases, food safety and foodborne diseases. Dr. Karun touched on this a little bit, the environment, and the functioning of health systems in the prevention, management of infectious zoonotic diseases, production-related diseases. We have a lot of them in the aquaculture environment, AMR, food safety, and other hazards. Okay, so let us look closely at the disease situation in the aquaculture scenario. So this, you are familiar with many diseases affecting aquatic organisms, finfish, shrimp, seaweed, mollusk, and there are many types of finfishes. And this has been our observation throughout the last 30 years. There is usually a long time lapse, typically years, from the time that there was field observation of an unknown or emerging disease, to its diagnosis, to global awareness, to the establishment of reporting system, surveillance, and cost-effective risk management measures. So I'll just give you a brief global distribution of the 3 diseases that Mohan mentioned. So this is how it looks like, how the epizootic ulcerative syndrome, also known as mycotic granulomatosis in Japan and red spot disease in Australia. So the data sources for these are WOA notifications, scientific and gray literature, technical reports, and industry information. Another classical disease of shrimp is WSSV, and you will see here the distribution. This data shows the first report from 39 countries and provinces, same data sources. Okay, another disease is AHPND, and this is the global distribution from the first time that it was reported in 2010 until the latest in Japan, in Okinawa. Okay? So, looking at the disease scenario in aquaculture, we tried to analyze what are the pathways, factors, and drivers on disease emergence in aquaculture. And out of this analysis, we created a causal web that is shown here. So from the many factors, drivers that we came up, we categorized them into 4. One is trade in live aquatic organisms. Second is knowledge of pathogens and hosts. Third is aquatic health management and control. And fourth is ecosystem change. When we analyze the many factors to disease emergence, we always put it in the context of the what we call as epidemiological triad that involves the interaction between the host, the pathogen, and the environment in disease development. So you will see here that there are 2 pathways. One is the exposure pathway that exposes the host to new or exotic disease. And it could be trade, new pathogen emergence, husbandry practices such as live feed, and wild population. If you look at the right side, these are the stress-related factors. And this could be changes in management, practices, soil and water chemistry, and so on and so forth. So these are stress-related factors that increases the impact to endemic disease. Now, the consequence of disease emergence is very much dependent on 2. One is the time of detection, and the other one is the effectiveness of response. And in turn, this will very much depend on surveillance capacity, availability of diagnostics, institutional coordination, research infrastructure, level of biosecurity, and options available for control. I'm stuck here. Okay, so having understood the diseases and how fish get sick, I would like to show you the economic dimension. So from the literature, we will find these losses. I think I captured Andy Shin's data here. But what does this show us? Numerous unmanaged disease outbreaks with high economic losses reflect an immature aquaculture industry. A maturing aquaculture industry, in the absence of any other biosecurity measure, requires a focus on disease prevention, and particularly governance and understanding disease impacts in terms of burdens and investment. I'm happy to report that aquatic sector is now engaged with the global burden of animal disease. This is a big program, GBAD, that is led by University of Liverpool. So the current approach to disease challenges, they need to be implemented by an economic dimension for improved responses and more efficient resource allocation. Okay, so now this brings us to this initiative that FAO and partners developed during the last 5 years. It's called Progressive Management Pathway for Improving Aquaculture Biosecurity. It uses the principles of being risk-based, progressive, and collaborative, again, within the context of this UNESCO circle. It has 4 stages. As stated here, stage 1, biosecurity risk defined, stage 2, biosecurity systems initiated, stage 3, biosecurity systems and preparedness enhanced, and stage 4 is sustainable biosecurity and health management systems are established. So what do we expect from PMPAB? We expect a reduction in the burden of disease, improving health at farm and national levels, minimizing the global spread of disease, optimizing the socio-economic benefits from aquaculture, attracting investment opportunities into aquaculture, and achieving One Health goals. Okay, so one of the instrument that I wanted to emphasize this, and this is a basic requirement in the development of the PMP, is what we call as National Aquatic Organism Health Strategy. This was advocated by FAO in 2007, but it gave new meaning in the development of the PMPAB. So you will see here all the technical elements that are essential and important to any aquatic animal health protection program, from policy legislation to aquatic animal welfare. You will see here that they are color-coded because it shows the stage of the PMP where this technical components should appear. Okay, so I have mentioned already the 4 stages. 2 important considerations in this PMP is risk assessment and emergency preparedness, always present at every stage. And each stage has key indicators and activities. I think there are 74 indicators. And there's what we call biosecurity action plans that serve as gateway passes to move from one stage to another. And as in any new framework, we need to develop tools in order to support its implementation. So we have 2 e-learning courses on risk, pathway to aquaculture biosecurity, managing disease risk in the value chain, and emergency preparedness for aquatic disease outbreaks. This is a free course, and you receive a badge. So you just Google FAO e-Learning Academy. Okay, the other toolkit that we have developed is this 12-point active surveillance checklist. And what I wanted to emphasize here is that this point number 12 puts surveillance in the bigger picture, which says that it should align to One Health goals and the Sustainable Development Goals. Okay, now going back to, I think almost all the presentations and papers, you will see it states that this is a very big challenge to aquaculture. Some will provide figures here and there. But I couldn't find, I did a very exhaustive research of the literature. I couldn't find a paper that tells me how to systematically assess until I got engaged with the GBAD people who are now developing methodology for assessing the burden. And this, I just highlighted these 3 papers. One is from Hong Kong, and the other one is 2 from WOA, authored by Ed Peeler, and they are introducing the GBAD methodology and application to livestock, to aquatic animal. But I'm happy to announce that we also have another paper that was recently published. This is Bridging Aquatic Organism Health and Economics in the Analysis of Disease Impact. So what we did in this paper is we got the methodology from livestock burden of disease, and at the same time, we also assessed the value of biosecurity using 4 hypothetical studies. And the outcome of that paper is that there should be more case studies, so I invite you to go to this paper. Whatever it is you are doing, whether you are developing a strategy, vaccination, diagnostic, etc., it might be useful now to do a CBA, and we have already the framework. Okay, I have 40 minutes. This is very important when you deal with action with AMR. Every country has a national action plan on AMR. You can, to check whether you have a national action plan on AMR, you have to go to the WHO website. But what is important for me is to see whether this national action plan has an aquaculture or aquatic component. So we did a review of 95 national action plans, and then we cross-referenced it to the countries, and you will see here that most of the top 10, with the exception of Egypt and Ecuador and Brazil, have aquatic component of AMR. I think I will stop here because I don't have time, but just one slide. This is, we are finalizing a book. We talk about zoonosis, no? So FAO commissioned a group of microbiologists, and we came up with some 40 species of bacterial diseases using these three criteria: economic importance, socioeconomic impact, and zoonotic potential. And out of more than 40 species, these highlighted in green are the ones that has zoonotic potential. Oh, can you give me one slide? Okay, I just wanted to say that I started working on fish health in 1986, and my very first publication is on trichodyna, okay? Because that was our thinking at that time. We are bug hunters. If you are a bacteriologist, you will look for bacteria. I was trained as a parasitologist, so this is my first publication on trichodyna, and from that simple skill of identifying parasites, we are now in this community of One Health. Don't forget, fish is here, we are important. And I just wanted to emphasize the logo of Fish Health section, somewhere here. It's this trichodyna. According to Dr. Arthur, this is the most beautiful parasite. Andy will agree. Okay, so I think this is my last slide. Whatever you do, do it wisely and think of the consequences. And while we are free to choose our actions, we are not free to choose the consequence of our actions. Thank you very much for your attention.
Sorry, a small technical hitch. So, Melba, thank you so much for a very info-rich talk. We're going to, I'm sorry, Melba, we're probably a little bit tight on time, so we're going to move on to our next speaker. But if you have any questions for Melba, please bring those into the coffee time as well. So now it gives me great pleasure to invite a very dear friend and our next speaker, Victoria Alde-Sanz. Just to give you her biography, Victoria has an MSc and PhD from Stirling University. She's worked for over 35 years in diverse aspects of aquaculture biosecurity. She has collaborated as an expert for international organizations such as the FAO, for the EU, for the European Food Safety Authority, for the World Bank and OIE, as well as with the private sector worldwide. She's published over 40 papers in peer-reviewed journals, over 50 articles in industry magazines, 5 book chapters, and Victoria is also the co-author of the CD-ROM Diagnosis of Shrimp Diseases and the editor of the Shrimp Book 1 and 2. And I must say these are very useful references. Very importantly, Victoria developed shrimp-specific pathogen-free stocks from WSSV-tolerant shrimp, which we used to recover the production in Saudi Arabia after the wipe-off of the shrimp industry due to white spot virus. So today, Victoria will talk on a call for further research on epigenetics with examples from the field. So please join me in welcoming Victoria to the podium. Thank you.
Good morning, everyone. First, I would like to thank Dr. Kuldeep and the organizers for inviting me to this presentation, to this conference. It's really one of the greatest conferences we have in the industry. Well, you know, I'm coming from research, but for the last 20 years I've been in farming, so I feel quite of an intruder here in this conference if it was not for all these good friends that I have. So what I'm bringing today is just some examples, observations from the field that we have the feeling that are related to epigenetics, and we would like to have a better understanding about them. So the starting point of this thinking was a decade ago, I read a paper about the effect of starvation in a pregnant woman and how it affected the descendant and how it affected also the next 2 generations. So at that point, we thought, okay, if that happens in humans, that's going to happen also in aquatic animals. So our thinking when we focus on production was that early life exposure, including the embryonic development, can, through epigenetic programming, determine the response to nutritional signals, immune response, and performance later in life. So the idea is that, if we could have targeted epigenetic modulation, we could optimize productivity. Yeah, and if we understood the heritability of these changes, it would be one more tool in terms of improving production. So I'm going to present just a few examples. Some of them are positive impact of what we think might have an epigenetic component and what are negative impact. So this is an example of broodstock that was raised inside the nucleus breeding center from post-larvae to adult size, and we compare the performance in the commercial maturation when we use 2 different feeds. We raise them from post-larvae to adult with a hyper-formulated diet, let's say higher protein than in the farms, high axanthancin, high in omega-3, and the other group, we fed them with the normal farm feeds. And then 60 days before they moved to the commercial production, for production of eggs and nauplia, we feed them with fresh feed. So we thought, during those 60 days, they will accumulate enough nutrients to perform similarly to the ones with the hyper-formulated diet. However, we see that the animals that were fed with farm feed, they had less spawns, they have less eggs per spawn, and they have less nauplia per spawn. So there was something about, the feeding during their cycle that affected the performance of as a broodstock. Another example that we performed in the farm in Aqua is the heat shock treatment. Peter Bossier and his group in Ghent University, they published a paper on the protection of, protection in Artemia through heat shock treatment against Vibrio campbelli. And they showed that, by exposing the Artemia to high temperature in several occasions, they could have a higher rate of survival. And this protection was carried on for 3 generations. So we just adopted this protocol for shrimp. We exposed the shrimp to 39 degrees for 2 minutes for 6 consecutive days. And then we had the support of Dr. Suparat and Kalaya, and they look at the expression of 5 different genes which are related to the immune expression. So we could see that gene ALF2 had an early induction after the exposure to the heat shock. We have 2 other genes, including heat shock protein 70, that it was a maintained response, and we have other 2 genes that had a high response. If you look, it is a much higher expression. So this protocol was adopted in a routine practice in the farm. So every future broodstock was exposed to this heat shock, and we had the hope that this is expressing this overexpression of immune response is expressed in the next generation. Another example is the way we raise post-larvae from PL4 to PL14. We had 2 systems. The normal commercial production is transparent water at a relatively normal density, 100 PLs per liter, and we were raising also the same stages, the same genetics, in biofloc at very high density, 260 PS per liter. So you see that the ones at high density, it was a bit smaller, and the survival was not so good. We never managed, we tried for several years, we never managed to increase the the survival. However, if you look at the size dispersion, the one in high density is very low compared with the one with the transparent water. And if we look at the growth in the pond in grow out, you can see in orange, this is the larvae that was grown in high density biofloc. In light blue is the one that was raised in transparent water. So even if we started with a smaller shrimp, that early feeding, maybe with biofloc or whatever conditions we had, it gave a much better performance in the pond. And in dark blue, what we have is the growth of the larvae that were stocked in a nursery, and then from there to the grow out. So even if, the grow out, the one that went through the nursery, it was a much bigger shrimp. Yeah, it was at least 2 weeks older. Still, you see the growth is much slower. And if you look at the coefficient of variation of these juveniles, it's much higher. It means that we were not raising them properly in the nursery, high density, not proper feed management, etc. So in a farm, it's difficult to decide which is the larvae that is performing better. Is the one that has higher survival at harvest, is the one that has higher growth, lower FCR. So what we look at it is the one that has higher profitability. So we measure it as a Saudi riyals per hectare per day. So you can see that the larvae that was raised with biofloc, high density, it had a profitability similar to the one raised in nurseries and then grow out. And it was much higher than the one raised in clear water. So if we can develop a system raising intensive biofloc, you can see the difference in the profitability. It's just 2.6 times higher density, so we need less infrastructure. We don't need to buy nurseries, yeah? And we get the same profitability that if we do all that investment. Now I have some negative effects of management. So in Saudi Arabia, we were at white spot in 2011-12. At that time, we tried different strategies of white spot that have been developed in Thailand that was basically exclusion, treating the water, PCR, craft fences, etc. It didn't work because our infrastructure, it was not suitable. It was like in Latin America. So there was no way it would work. So if we look at Thailand, Thailand immediately started with SPF strategy, PCR negative, etc. It took them 4 years to recover the production, and then when they switched to SPF Vaname, it allowed the exponential growth. In Ecuador, as I said, they have a similar infrastructure to Saudi Arabia. There was no way to exclude the pathogen, so they just lived with the virus. It took 7 years to recover production. But the good thing about it is that the shrimp naturally developed tolerance to the pathogen. So in Saudi Arabia, what we did was a combination. We used the SPF approach of Asia, and we used the tolerance that had been developed in Ecuador. So for that purpose, what we did is to develop a population that was SPF and that was SPT, there was tolerance. So we selected this population in Ecuador, and we did just the opposite of what is the traditional SPF development. Instead of going to remote areas to find clean shrimp, we went to farms where there was epidemics, there was mortality. We collected big shrimp, 25 to 30 grams. We exposed them to certain stress, and then whatever was PCR negative, we kept it for this population. So at the end, we have a new product that was SPF, SPT, or SPR. At that time, we did this in 2010, and it was based on what was the hypothesis, accommodation hypothesis of Tim Flegel. Nowadays, it's not a hypothesis anymore. It is proven. Suparat has been talking about that, the presence of EVEs in the shrimp genome that confers this tolerance to the animal. At that time, there was a lot of confusion on the terminology, what is SPF, SPR, SPT. So FAO funded a small workshop and then a publication to define all these terms. Okay, we brought those animals to Saudi Arabia. Initially, we didn't have a challenge facility, so we didn't dare to select for better growth. we thought that, if we select for better growth, we may be selecting against tolerance. So it took us a few years to start the improvement programs. So once we have the facility, we expose every cross on triplicates and to look at the survival under a standardized challenge set. And over the years, we have improved, we have put more pressure in this selection. And instead of looking at high survival at 8 days, we do it at 14 days. And we are also taking into account the viral replication rate as it relates also to infectivity. So our farmers wanted more growth because if we compare our shrimp with the ones in Southeast Asia, we realized we were growing very slowly. So first we compare the fast-growing shrimp tolerance to white spot to our own tolerance. So you can see this is the Saudi shrimp, this is the fast-growing shrimp. Huge difference. And you must remember Saudi Arabia has extreme weather conditions. You can have 10, 15 degrees difference between day and night. So it is, the first thing you want is that the shrimp survive, and then you want the growth, but you cannot risk to have a better growth if they don't survive. So what we did, we created hybrids. As you can see, the hybrids, they lost tolerance. So from there, we started selecting, and you can see the result. This is 2019 survival of the hybrids, and this is 2023. So when we felt confident that the animals were already tolerant enough to go into commercial scale, we moved them to the farms. And right now, we have 3 genetic lines, and the only one that is used commercially are these hybrids. So we know we have a genetic tolerance to white spot. This is a work we are carrying out with Dr. Kalaya, Dr. Tim. We have these ifs, they have been identified. We are trying to understand how they are inherited, how this is expressed, and so on. But the genetics doesn't solve poor management. So you see this is our first outbreak of white spot. Once we introduce the SPF, SPT, the production recovered in an exponential way. However, we really abused the system. We built nurseries, we did hyper-intensive, not enough water exchange, and you can see ammonia levels. It should not be above the red line, and it goes up to 12. And there was someone presented here the susceptibility of white spot, in high ammonia, yeah? And it was very clear. So we had severe mortalities in the nurseries. So even if we have a genetic tolerance to white spot, this expression of tolerance was silenced during this period. Yeah? So the moment we pushed the animal back into the pond, there was very little difference between positive and negative ponds. So the animal recovered that tolerance during that time. But that tolerance was not there while we had the stressful conditions. Another example is the importance of artemia. At one point, we were short of artemia, so someone decided, okay, let's save money. In Ecuador, they can raise post-larvae without artemia, and then you can see the size of the post-larvae at PL14 at harvest, depending on the number of days they have received artemia. It's a huge difference. And then these animals, we moved them to nurseries. You can see the survival in nurseries, very inconsistent, poor. In the pre-growth, which is another system of nurseries, again, the same problem. Or when we move these animals to the grow-out phase. Yeah? So the importance of the artemia in an early age, yeah, it really reflects up to the end of the cycle. Our farmers, they will say, okay, it's not important to have low survival in the hatchery because then we select for the strongest animals. Yeah, but that is not correct. Yeah? So you can see here, we trace the tanks, the survival in the tanks in the hatchery with the profitability of those post-larvae at harvest in the grow-out pond. And you can see it's directly related. Yeah? Of course, there are some variables. Sometimes you have power shortage, you have other variables, but there is a very clear tendency. The moment we have poor survival in the hatchery, the profitability of those animals goes down. So we need to understand, what is the minimum survival to make money in grow-out? Let's say, is it 50%, 55? Another example, they said, okay, no problem to have poor quality PL, we put them in a nursery, and there we filter out the weak ones, and we have the stronger ones. But that doesn't work as well. Again, this is the survival in the nursery, yeah, and the profitability of those animals at harvest. And you can see, you know, the moment your survival goes down, the profitability in the pond goes down. So those early age, the care of those animals in the early age is very important for the final performance. That is just an example. This is the different farms in Aqua. You know, they are one next to the other, and these are results, the same cycle, so it's the same genetics, it's the same weather, it's the same infrastructure, it's direct stocking, and you can see the difference in terms of profitability within the same farms. Yeah? So in red is the ones of the post-larvae raised with biofloc, high density, and in green is the one of clear water. And you can see the difference between the two types of larvae, but especially you can see the difference between the farms. Yeah, huge difference. And, you know, every year, these two farms, they perform the best. And these are the two R&D farms. It means that those are the farms where we are on top of them. We are always watching them. It's about caring. It's about prevention. It's about looking at your oxygen. It's about the Seki disc and so on. It's being on top. We cannot always blame, oh, it's bad genetics, or feed is bad. No, no, no. You need to analyze how you can improve your management and be on top of that. A quick comment on epigenetics and breeding programs. We see a lot of research on genetics, genetic markers, and so on, but we feel that we need more information on epigenetics. We believe that the breeding programs cannot be properly run without the understanding of epigenetics influence. And we think that husbandry is the main component of a breeding program. We need to integrate the cultural conditions, the productivity parameters, and need to be considered for the estimation of the breeding values and the accuracy of the estimations. Yeah. So in summary, you know, we need a better understanding of the factors in early development, including gametes, shaping epigenetic differences. So in terms of management, cultural conditions, temperature, nutrition, feed management, treatments, chemicals, vaccination, bacterial exposure, the functional genomic changes that occur in the response to these epigenetic changes. We also need to understand the heritability or reversal of these epigenetic changes. And also, we need better understanding of the relationship between epigenetics, phenotypic variation, fitness, and productivity. So in the meantime, before we get all this information, the ball is on our camp as farmers. What we need to do is to optimize cultural conditions. Look for higher survival, look for lower coefficient of variation, look for lower FCR. It's all about welfare. Whatever you want to call it is welfare. And before I finish, I wanted to raise your attention on this training course. It's organized by Biotech under Dr. Kalaya, and it's an excellent opportunity for farmers to learn about diseases and good management, and also for researchers to learn about farming. So it's an excellent platform for interaction between farmers and scientists. And that is all. Thank you very much.
Thank you very much, Victoria. We have time for one quick question. If we have, yes, Celia.
Thank you very much, Victoria, for going us through your years of experience from a failing farm to a successful farm. But my question is mainly on, there is a move to stock post-larvae coming from non-ablated females. How do you think the hatchery would be able to deal with that? Would we be able to produce synchronously millions of nuclei the way we did with ablation? And what is the alternative?
Yes. Well, I don't have the experience for it, but I can argue that, you know, it would be better without the ablation. But however, when you perform the ablation and you do it well, the animal gets back to feeding within 1 to 2 hours. So the stress of the ablation, properly done, is very little. Yeah? So I don't know. At the end, it's all a matter of economics. If you need to duplicate your number of broodstock, the facility, farming at the end is economics. It needs to be profitable. But I don't have the experience to develop a protocol because you need to adjust the feeding, you need to adjust the rate male, female, and I don't have the experience for that. But I only want to say that the stress of ablation is not so severe. An animal will not go back to eat within an hour time if it was, let's say, stress or damage. Yeah? The feeding is so important. You can really see the health status of your animal by the feeding behavior.
Yeah, but I think the issue is like the ablated female loses one eye.
Sorry, say it again?
Ablation makes them lose the eye.
Yeah.
So it's the welfare issue of that.
Yeah.
So, I don't know. It's a big problem coming up. Thank you, Victoria.
Thank you so much to talk about, but thank you very much.